My name is Savannah Yates. I'm a senior exercise science student at the University of Indianapolis, and today I'm going to discuss whether the microvascular oxygenation response is related to handgrip muscle endurance. Near-infrared spectroscopy is a non-invasive measure of microvascular tissue oxygenation. It is measured by placing a sensor on the muscle. In our experiment, this is electrodigitorum superficialis, where near-infrared light is emitted into the tissue, and it changes wavelengths as it is reflected back into the sensor based on whether or not oxygen is attached to hemoglobin. The nearest recovery response is how quickly oxygenation returns to baseline post-exercise. This is linked to PCRB synthesis and is used as an index of skeletal muscle capacity. Past research has suggested that slower nearest recovery response is linked to lower oxidative capacity of skeletal muscle and that recovery can improve with cardiorespiratory training. Previous research has also focused mainly on the recovery response following cardiorespiratory exercise. However, obtaining this data following submaximal isometric exercise may be of interest because there's less noise affecting the signal, and it can be used with a wide range of populations, including clinical populations. The purpose of this investigation was to assess the potential relationship between muscle endurance and NIRS response during and immediately after submaximal isometric contractions. It was hypothesized that there would be a significant relationship between exercise SCO2 and muscle endurance, as well as SCO2 recovery and muscle endurance. In this study, we focused on hand grip, where our subjects performed submaximal and maximal isometric contractions using a hand grip dynamometer. Our subjects were seated with their elbow flexed at 90 degrees, supported by an armrest with their wrist in neutral position. Muscle oxygenation was collected using a NIR sensor placed on the flexor-digitorum superficialis of their dominant arm, sampling at a frequency of 2 Hz. The participants in this study included 15 total participants that were healthy college-age students. This graph showcases a drop in tissue oxygenation that occurs at the onset of exercise, that is labeled as end-exercise SCO2, and their post-exercise recovery response, which is measured in half-time, that is the blue line. Figure on the right outlines procedure. The first participants performed 5 seconds of a maximal voluntary isometric contraction. There were 3 trials total with 2-minute rest in between. We averaged the 2 closest trials to find their MVIC value. Following this, there were 2 identical sets of submaximal isometric contractions at 25, 50, and 75% of their MVIC, with 1-minute rest between each of the 5-second contractions, a 6-minute recovery between sets, and a 10-minute recovery following both sets. After this, subjects performed static endurance assessments, where they held a 50% contraction until volitional fatigue. Their static endurance value was calculated to be their force multiplied by the time they held the contraction. The final assessment was the dynamic endurance assessment. Here, participants performed 12 3-second maximal contractions with 5-second rest in between each. To calculate their dynamic endurance, the average of the subject's last 3 contractions were divided by the average force of the first 3 contractions, multiplied by 100 to find the percentage. Values obtained from the NIRS data were exercise SCO2, which is the minimal SCO2 during contraction, and half time, which is the time to recover from the minimal value to half of their peak value. The statistical analysis performed were intraclass correlation coefficients to assess within session reliability of NIRS measures between set 1 and set 2 of the submaximal isometric contractions, and Pearson correlations that assess the relationship between NIRS measures and hand grip muscle endurance measures, with the statistical significance at set as p less than 0.05. Pictured in this table are each of the ICC values from when set 1 was compared to set 2 for submaximal isometric contractions. Below this value are the 95% confidence intervals and whether or not the ICC value correlates to a good, moderate, or poor reliability. Each of the values that have an asterisk next to them are statistically significant, with our p-value of less than 0.05. From the statistically significant data, we ran Pearson correlations between the minimal SCO2 values and the half-time values compared to the static and dynamic endurance values. Results from this analysis showed only one statistically significant correlation between exercising SCO2 value and static muscle endurance, which is a moderate correlation shown by the R equals 0.569. So overall, we found that 25% and 50% provided a moderate and good within-session reliability for identifying SCO2 recovery. However, there were no correlation present between SCO2 recovery and static or dynamic muscle endurance. In the 75% MEHTODS, it was found that there's a good within-session reliability for identifying exercise SCO2, and a moderate correlation exists between exercise SCO2 and static muscle endurance. This means that these individuals are more economical and they did not use as much oxygen during exercise to have a higher static muscle endurance.